Hi, my name is Keretz, and today I'll be talking about ecological niche modeling for native and invasive mussels in the St. Clair and Detroit River systems. To start, native freshwater mussels, or unionids, are a macroinvertebrate bivalve that contribute multiple ecosystem services to our environment, such as water filtration, nutrient cycling, and sediment stabilization. North America hosts the highest diversity of unionids worldwide, with approximately 300 species across the continent. And in the Great Lakes region, there are approximately 43 species in Michigan and Ontario. My study area, the St. Clair and Detroit rivers, are in the Huron-Erie corridor of the Great Lakes. These rivers form the international border between Michigan in the US and Ontario, Canada. Additionally, these rivers are part of the Great Lakes Shipping Channel, and they historically supported 39 unionid species. However, unionids were considered extirpated from these rivers shortly following the introduction of zebra and quagga mussels, collectively Dracenids, into Lake St. Clair in the 1980s and the 1990s, respectively. Fortunately, there is hope. Unionid refuges have been found in the Lake St. Clair Delta and in Lake Erie coastal wetlands. And due to their highly imperiled nature, it has become increasingly important to identify and protect the unionid populations in these spatial refuges. One method for locating unionid refuge space is modeling known refuge locations. In 2018, Bostonbrook et al. Were able to do just that, where they successfully predicted unionid presence in Lake Ontario using Maxent, unionid presence locations from Lake Erie, and variables like fetch, shoreline geomorphology, and depth. Following up to this study, we are asking whether mussel presence can be similarly modeled in large river systems, particularly the Detroit and St. Clair rivers. This leads me to our study objectives. The first component of this study is to survey the Detroit River for Dracenid and unionid mussels. We will then use this data to create predictive species distribution models for both Dracenids and unionids to predict presence in the St. Clair River. Finally, we will then survey the St. Clair River to validate and assess the predictiveness of the Dracenid and unionid models. So we'll start with Dracenids and cover unionids in the second half of the presentation. For our methods regarding Dracenids, we sampled the Detroit River in summer of 2019. At each site, we collected 6 petite ponar grabs, and any Dracenids found in these ponar samples were identified to species and quantified. In the Detroit River, 35 sites had live Dracenid mussels. This map shows the estimated densities, where the small black circles represent sites with 0 live Dracenids per meter squared, and the largest circles represent sites with over 5,000 live Dracenids per meter squared. The dark blue represents live zebra mussels, and the lighter blue represents live quagga mussels. Sites in the St. Clair River with live Dracenids present ranged from 7 to 6,341 live individuals per meter squared. Now I used our Dracenid present sites and Maxent, which is a presence-only predictive modeling software, to create a species distribution model for the Detroit River. The variables I considered were depth, total velocity, east-west velocity, north-south velocity, and river distance to the river's inlet, outlet, and nearest tributary. Within Maxent, I split the presence data into 80% training data and 20% test data, and each model creation went through 10 replicates. Additional settings were based on these previous papers. Finally, all models were assessed using the area under the curve, or AUC, a number that ranges from 0 to 1. A general rule of thumb is that an AUC of 0.5 means the model is no better than random prediction. So here's the Dracenid species distribution model for the Detroit River. This model used 4 variables: depth, east-west velocity, and river distance to the river's inlet and nearest tributary. This model has an AUC of 0.749. And as shown by this map, the red coloration represents a high probability of Dracenid occurrence, and the green coloration represents a low probability of Dracenid occurrence. I then overlapped the sites where Dracenids occurred to estimate occurrence probabilities for each site based on the model. Among all the Dracenid present sites, the occurrence probability ranged from 27 to 99%, and the mean occurrence probability was 67%. Finally, a majority of our Dracenid present sites had an occurrence probability that was greater than 50%. Keep that 50% in mind as we'll use it to assess future models. Next, I projected the Dresenid species distribution model to the St. Clair River, and it predicted a high probability of Dresenid occurrence in the upper reach of the river. To validate the model, we surveyed the St. Clair River in summer of 2021, using the same methods that we used in the Detroit. Of the 51 sites surveyed, 40 of them had live Dresenids. As shown by this map, where once again, the small black circles represent sites with 0 live Dresenids per meter squared, and the largest circles represent sites with over 5,000 live Dresenids per meter squared. Sites in the St. Clair River with live Dresenids present ranged from 7 to 5,928 live individuals per meter squared. Now, using the sites where Dresenids were present in the St. Clair River, we can assess the projected model. Please remember that a majority of our Detroit River Dresenid present sites had an occurrence probability that was greater than 50%. Well, 36 out of the 40 St. Clair River Dresenid present sites also had an occurrence probability that was greater than 50%, meaning that the model predicted live Dresenid presence among sites where live Dresenids were found with 90% accuracy in the St. Clair River. That brings us to the overall goal of this work, which is to facilitate our understanding of Dresenid distributions in both the Detroit and St. Clair Rivers more than 30 years post their invasion. This understanding could potentially assist with recovery efforts of impacted species, mainly native mussels or unionids. So now we'll be switching gears over from Dresenids to unionids for the rest of the talk. To collect our unionid data, we surveyed the Detroit River again in summer of 2019. Sampling for unionids was done by a 1 person-hour time search using scuba, and any live unionids found by divers were brought to the surface, identified to species, and quantified. We surveyed 56 sites throughout the Detroit River, and we found live unionids at 5 of those sites, represented on this map by the yellow circles. Among these 5 sites, we found a total of 220 live unionids, representing 11 species. Now I used Maxent and the sites where live unionids were present to create a species distribution model for the Detroit River. The variables I considered were the same as those previously described for Dresenids, though I also used the Dresenid species distribution model to represent an estimation of Dresenid presence throughout the river. Within Maxent, I used the same parameters and methods as were used for Dresenids to create the unionid model. So this is the unionid species distribution model for the Detroit River. This model used 5 variables: depth, total velocity, river distance to outlet and nearest tributary, and estimated Dresenid presence. This model has an AUC of 0.956. And as shown by this map, the red coloration represents areas where unionid presence is likely, and the blue coloration represents areas where unionid presence is unlikely. I then overlapped the sites where unionids occurred to estimate occurrence probabilities for each site based on the model. Among the unionid presence sites, occurrence probability ranged from 41 to 97%, and the mean occurrence probability was 77%. Finally, 4 out of the 5 unionid presence sites had an occurrence probability greater than 60%. Again, keep that 60% in mind as we'll use it to assess the St. Clair River models. I then projected the unionid species distribution model to the St. Clair River, and small pockets were highlighted as places where unionids were likely to occur. To validate the unionid model, we sampled the St. Clair River in summer of 2021, using the same methods that we used in the Detroit River. We sampled 51 sites in the St. Clair River, and were fortunate enough to find live unionids at 7 of those sites, represented on this map by the yellow circles. Among these 7 sites, we found 14 live unionids, representing 9 species. Now, using the sites where unionids were present in the St. Clair River, we can assess the projected model. Please remember that a majority of our Detroit River unionid presence sites had an occurrence probability greater than 60%. Well, unfortunately, 0 out of these 7 unionid presence sites had an occurrence probability greater than 60%. In fact, the mean occurrence probability for these 7 sites was just 7%. So now in big, bold letters, the model created from the Detroit River data predicted live unionid presence in the St. Clair River with 0% accuracy. All right, that asks the big question, why? Well, my first step was to take these unionid presence sites and create a new species distribution model just for the St. Clair River. This model used 5 variables: estimated descendant presence, river distance to inlet and nearest tributary, east-west velocity, and north-south velocity. It has an AUC of 0.949. Now I'll compare this model to the Detroit River model. So here's the unionid species distribution model created for the Detroit River and the variables used to create this model. Now the major contributing variable to this model is total water velocity. Now compare this to the variables used to create the St. Clair River model, where the major contributing variable is estimated descendant presence. So the main contributing variables for these models are not the same. So it makes sense that the models are currently unable to project well to the other river. This tells us that the unionid models made so far are highly predictive for their respective rivers, but do not predict well in other rivers, at least not yet anyway. This is strongly contrasted by the descendant model, which was highly predictive for descendant presence in the St. Clair River and could potentially be used to predict presence in other large river systems. With that, I'd like to thank our funding sources and all of our collaborating agencies. This work does take a small army to accomplish, so I'd like to thank every person on this list for being amazing and for their continued dedication to this project. And if you have any questions or comments, please feel free to reach out. You can also follow myself and my advisors on Twitter if you would like to keep up with this work and the other projects that we have in the Great Lakes region. Thank you for watching.